So first of all, obviously, like to thank Philippe that I'm here. Even more so, I'd like to thank him that he gave me opportunity to work in Singapore, because he and Paola were able to set up an institute which has created an environment which gives opportunities to work on topics which you definitely cannot do anywhere elsewhere. So one of these things, as you can see, is a bit esoteric, genetic quantum states, which I will hope to end with. So, to begin, however, I wanted to start with the subtle traits of regulation, and to very much begin with is basically the topic of that meeting, and that was the question from mice to men to bench to bed, and the question is, can we really study complex human diseases in mice? And my personal opinion is actually no, and I have two reasons for it. One is this: mice and men are indeed quite different. Moreover, and that is let's say some kind of knowledge which came to me more in the recent time, is that all mouse models are totally bred into complete homozygosity, which means bulb C, as it's written there, don't have any SNPs anymore. So, does it matter? I think afterwards you will think yes. So, to clarify one thing, what is a SNP? A SNP is a single point mutation, but not any. It has to be there at least in 1% of the population. So as a consequence, it happens roughly every 100 to 300 bases, so that the total of SNPs at the moment identified in the recent 1000 Genome Project is something like 15 million SNPs. Of these ones, about 70,000 are coding, and these are what most people are interested in, because they make amino acid changes and frame shifts and ruin genes. And in fact, about 2,300 loss of function genes have been identified, which is actually 10% of the genes. Moreover, in about, in every one of us, about 1% of the genes are knocked out, which is 250 to 300 per individual. It's quite a number. You can only hope it's not the important one. So, since everybody's working on that, we don't, because when you subtract 68,000 from 15 million, there are 14.9 million SNPs left. And these are indeed what I think the way more interesting one, and these are the non-conic one, because they affect intergenic, promoter, and intronic region, and the effect is way more subtle, in a way that usually what it does, it affects quantitative expression of the gene in form of affecting transcription factor binding site, splicing sites, enhancer sites. So one more thing what I want to say before: in order to do EQTL analysis, and also for other things, we developed an SIGN, a systems biology system, which is extremely powerful. It's basically it's a cohort study or a cohort-based model in which we have 500 individuals, healthy Chinese, and these ones are absolutely in-depth characterized. We have their clinical and social status. We have their genome data, so we have the 5 million SNP data of them. We have transcriptome data, complete one, plus phenotypic data. And this phenotypic data is excellent because it's not done by just one group. Basically, every PI at SIGN almost was involved in characterizing its share. So, and all this data afterwards gets thrown into one joint integrated database. So what this tool produces, I hopefully can try or can convince you in the following 15 minutes or so. So let me start with one SNP, or better to say one gene, which came up out of the study. It's VSTIM1. It's an inhibitory receptor. It's a surface receptor with unknown ligand. So this is basically the genetic characteristic. It's a real QTL. Depending on the genotype of the SNP in the promoter region, you get a little bit more, or a lot of the thing expressed on the surface. As an inhibitory receptor with E-term motive, it recruits SHIP phosphatases and can inhibit a number of activating receptors, which all work with items. Example is shown here for the Fc epsilon receptor. So if you cross-link that thing, it can effectively shut down the Fc epsilon receptor signaling. From another group in Holland, it was already reported that this thing is myeloid cell specific, and it's found on neutrophils, eosinophils, and monocytes. So when we look at the position of the SNP, it's a single SNP here, very close to the transcriptional start. Most likely, it affects this transcription factor. I mean, since it's on myeloid cells, it has to be a myeloid zinc finger in that case, coincidentally. And this is sort of the soft spot here. That's where the polymorphism is, which lets the factor bind or not bind. So while this is a myeloid factor, one would expect that it affects the expression of that thing on all myeloid cells. To our surprise, it didn't. So here are 3 individuals with 3 different genotypes for the VSTIM1 promoter. And as you can see here, top 1 neutrophil, monocyte, lymphocyte shouldn't react. In all cases, the neutrophils were positive for VSTIM1, disregard of the genotype of the SNP, whereas here, monocytes reacted very sensitive. If you're CC, you're almost negative. CT become positive. TT, full positive expression for that marker. It's even more clearer here when we just go for CD14 positive cells. Here is the CC genotype, mostly negative. CT, the heterozygous, they just shift out. And fully positive also in numbers is here with the TT. So moreover, when we just overlay them like that, we basically have 2 populations which we find in any of the donors. The VSTIM low ones here, and the VSTIM high cells. Su Cheng did a deeper characterization of these 2 populations, and what she found is actually that they are not just negative or positive for VSTIM1, but for a lot of other markers. For instance, here, this is an interesting one. CLAG5A is a receptor for the dengue virus. But also the same thing, IL-13. The blue ones here, are the ones which are low, are usually low for IL-13 receptor. So with other words, what we have here are monocytes of 2 different flavors. And the interesting thing with the SNP is that it basically balances numbers. So here, the TT, we have a high number of the red cells. CT, the numbers decrease. And here, if you have the CC, you have mostly only the blue cells here. So in this case, this SNP basically balances between individuals the number of these monocytes. So a very similar phenomenon we also identified when we looked at the Fc epsilon receptor. So FcER1 is part of the Fc epsilon receptor complex and was described before as an EQTL, where on basophils, this SNP or the polymorphism on that SNP should control the regulation, and that way in a very simple way. Also here, the SNP is in the promoter region, here at position 66. And here, the polymorphism was described to affect GATA1 binding. Okay. So far the theory, published basophils, shouldn't be a problem, we thought, but when we got our Singapore basophil, there was nothing, nil. In 500 individuals, we could not see any of the described effects. It was a bit surprising. I mean, a little later turned out, when you go to the fine print of the papers, that in order to see this effect, they had to take the basophils and strip away the IgE. So, however, under natural conditions, there's IgE. Consequently, the whole polymorphism has no effect on basophils. It was a bit disappointing. So one advantage of our systems biology approach is that we cover a whole range of cells, not only basophils. And in that case, we could see that beside basophils, also PDCs and myeloid disease are positive for the Fc epsilon receptor. So looking again at these data, now for these cells we found, yes, here's something, that PDCs, in fact, are targeted, are affected by this polymorphism. In PDCs, the switch from CT to TT indeed makes the expected increase, how it's described before for basophils. Myeloid disease, interestingly, don't care, nothing. So moreover, on top of that, in this, with these data set that we have, we can also do hypothesis-free analysis. And in that case, basically just trying to correlate the polymorphism with all the parameters which were put into the database. And what came out of that is, in fact, that the main target for that SNP is monocyte. This here with a p-value of e minus 6, e minus 7, you could clearly see that this SNP renders monocytes, which are normally negative, positive. This is nicely seen here. If you're an individual with a TT here, there's absolutely no Fc epsilon receptor on the blue cells, also no IgE. As soon as the C is there, cells get leaky and IgE can bind. So what is also interesting here is that the regulation, in that case, is opposite to what we see on the DC. The transition from CT to TT shuts down the expression, whereas on PDCs, it goes up. So this is just the overview for the 4 cells which are Fc epsilon receptor positive. We have nothing on myeloid cells. We have nothing on basophils. On basophils, most likely because it's compensated by the beta chain they are having. But we have opposite effects on monocytes and on plasmacytoid disease. So this means that also here, like before for this VSTEM1 effect, we have rather, instead of just a simple classical EQTL effect, we have a very nice balancing effect. And this one, in this case, actually goes towards the allergen response, because we're talking IgE here. If we start up with individuals which are TT, they have a strong PDC response. CT, this response goes down, the monocytes start to react, and presumably, we didn't have individuals, this is enhanced if the people are CC. So, while this one here balances the flavor or cells of different monocyte flavor, this SNP here is really balancing the response, monocyte versus PDC. So, another interesting aspect of these non-coding SNP is SNP architecture. This is something usually not considered because people think a SNP is a point. In fact, it's not. So on this, we did on another molecule we're working on since quite a while, CD39. Also here, we have a polymorphism which acts very strictly cell type specific. So you can see here, Treg cells in CD39 individuals which are CD39 low don't express it, whereas in donors, which we defined as CD39 high, they do. This is different to B cells because here the individuals don't care on this polymorphism. So in this specific case, we could pin it down to a SNP RS7071, which is again a promoter SNP. And this SNP sits here ahead, about 10K ahead of the ENTPD gene locus. But here it was very clear that this is probably not the causative SNP. And the reason is here that the whole gene region is very big. It's about 200 kb, and we have many, many, many SNPs, of which many of them are linked in some way. So this is a standard way to look at it. Every time when it's red, then this SNP and this SNP are in perfect linkage. Yeah. Well, this, this pictogram, or however you want to call it, gives some kind of a nice image. It's not very informative. So we transformed the whole thing into something like a hierarchical SNP structure. So if you do that, suddenly things become much clearer. In fact, instead of just having such a blurred red thing, you suddenly see here that all the SNPs, particularly the non-coding SNPs, are organized in clusters of perfect linkage. So this one and even these ones, where we have single SNPs, are linked to the main cluster so tightly, there's probably just one single individual was differently. So this was, for me, at least as an amateur entering the field, a big surprise, because I never expected such a level of organization. So for this SNP here, the 7071 SNP, which we used in a way as a tag SNP, we could say this thing matches to the first cluster here, cluster A. In principle, however, every single SNP here could do the trick. They're all linked. Moreover, the second cluster here, cluster B, is linked to the first cluster also with a factor of 0.82. And what I found totally amazing is that you can have two of these perfectly linked clusters which even overlap in that way. Come to that in a moment. So question is now, which of these SNP or which of these clusters is causative? On closer look here, one sees here two SNP, SNP A and B, these ones, which nicely flank the first exon of the CD39. This thing here offers a binding site for NF-kappa B, which is close to a FOX binding site, which is a thing obviously good for the Treg cells. The other one here is STAT3 site, which switches to a BRCA site. Moreover, when we look at Chinese, these two clusters are fused into one. So this is Caucasian and Chinese, and there these two SNPs are together and inherited together. So this, as a consequence out of that, one can say if we look at Chinese with this perfect linkage, we will have three functional states for CD39: low, intermediate, and high. If we now go to Caucasians, which have this two link clusters, suddenly the number of possibilities increases. We have two additional states here and here. So in order to define whether it's cluster A or SNP A or cluster B, SNP B, which does the trick, we just had to hunt down individuals here and measure them. The result, however, was it's both. So you can see here, even for this intermediate one, also their value for the CD39 expression is intermediate. So that practically really both clusters contribute here to the expression of the gene. And I personally think this is not a coincidence, it's probably like a common theme. What one also can see nicely here is that the effect of these non-coding SNPs, first of all, is very strong, basically from nothing to a lot, and moreover, the effect is strictly cell type specific. Treg cells are affected, B cells are totally not. So to wrap it up here, this is this architecture thing. SNPs, particularly the non-coding SNP, are really organized in clusters most of the time, which basically you can consider as slots, evolutionary approved mutations, which can hold transcription site. So I would bet that normally when you have a cluster, it's not the one factor, but many factors which bind and which can all either act complementary or synergizing on the expression. The second thing what you can get out of it is that moment when you have these overlapping clusters with a linked or with an imperfect linkage, suddenly you can realize more genetic states. Instead of having this 5-gear shift, if you want, like in Chinese, Caucasians can do fine regulation with 5 steps. So, networks. So while these things basically describe the interaction on the gene, on neighboring gene, and the interaction between SNPs, which are proximal to each other, we can obviously also have functional linkage. This is a thing which, again, is more specific for non-coding than for coding SNP, because usually when a gene is disrupted, there's nothing to compensate. Here, with these non-coding SNP, as soon as genes are affected which target the same trait or pathway, they interact either by acting additive in the case of the CD39 or even epistatic. So in that sense, these SNPs form networks very similar to the genes they are controlling, and the dosage, if you want, of these genes, or the allelic dosage, is simply dictated by the Hardy-Weinberg equilibrium. That's what the slider shows. And the purpose of these things is very trivial. It's this, it's for him. It's evolution. Instead of every time inventing a new SNP, you use the evolutionary approved ones. And the ratio, in this case, is basically already adjusted here in the Hardy-Weinberg equilibrium. So evolution is not making new, it's rather mixing. So you can also do the reverse. So if you have a set, a really clean set of GWAS data for a certain condition parameter, like in that case, rhinitis, you are able, by tracking down the epistatic interaction, to reconstruct the actual underlying pathway. So here we did it in that case for CD39. Admittedly, we have to say the data set was not so perfect, but it's good enough for the principle. So if you do an epistatic computation here, based on the polymorphism here, and identify all the genes which are in interaction with it, you come up with a list. It's actually not too many. And these genes you can put into a logic context, like that, a pathway. And this pathway is very clearly dependent on the condition you are using. Yes. The beauty of that thing is that whatever gene you identify is kind of the gene. So it's not that it says it's a protein kinase D, it tells you it's protein kinase D3. In that case, we could even prove it by showing that the underlying polymorphism for this protein kinase D3 is, in fact, an EQTL which behaves as expected. Could also found a few more things of this pathway, like here, the HDAC3, GATA3, and so on. Same thing up here. Here, it postulated to have this CATM2 as an epistatic partner. And also here, we could show that the expression is indeed dictated by the SNP identified by the approach. So, in a way, this gives me always a shiver when I look at it, because it tells you that from a piece of DNA, you are able really to deduct the truth. In a way, it's a matter of shining the right light. You can have the DNA from toenail or whatever. If you have enough, it's proper annotated. You just need the conditions to make it tell. Yep. So, lastly, to finish up, as promised, some quantum mechanic implications. So when I said that basically this network is simply dictated by Hardy-Weinberg, in a way it's only half the truth, because in reality, this applies only if you look for the population. So if you take this as a slider for the allelic state, which correlates to a certain phenotype, then this allelic state can be something between 0 and 1. However, if you now go to the individual, it's not. We have 3 defined states instead: AA, AB, BB. As a consequence out of that, you can only have 3 phenotype states. These ones here. So, and if one looks at this pattern, then it's very striking that it looks almost like that, like the quantum mechanic model. This is the transition from particle to wave. Also here, if you look for the energy levels on the macroscopic state, we have a continuum. If you go down on the microscopic scale, which sets it up, you end up with discrete levels, energy levels here, which defines the transition. And this all is mathematically described here from him, Schrödinger, and his famous equation. So actually on a trip to San Diego, I met up with Ginesta Bianconi. She's a professor at Northeastern and an expert in quantum statistics, especially in Bose-Einstein statistics, and she was able to actually use quantum statistic principles to compute or simulate the multi-Lotzi evolution of a diploid population. So here she started up, in that case, from the epistatic idea of having coupled pairs, and was able to get an accurate model, and even could describe a little bit the condensation, which at the end describes the extinction of a paired SNP. So, does that mean now that we would have, instead of quantum mechanics, have now something like quantum genetics? Unfortunately, obviously not. This is so far as just an expert a test, but to my feeling, it has potential. It's worth to be explored, because on one side, quantum mechanics offers possibilities which are simply before unimaginable. Things like tunnel effects is something which you cannot do in classical mechanics. On the other hand, it's also a bit scary, because if you go really into quantum mechanics, it tells you that everything you do in your physical world just depends that you can observe it. So, we'll see. That's it. So these are my people, my team, very proud to have them. Here, Mireille, Lydia, and Dilip and Anand. These are already since quite some time my lab. Forgot to have a photo. Collaborator in Berlin, Kirsten, and particularly this guy here from NUH, Van de Yoon, very important partner. And most important is really the people at SIGN, which not provide blood and samples, also provide data, interaction, and collaboration. And that's it.